Hello students, welcome to my lecture. In today's lecture, I will explain you sexual reproduction in the flowering plants, which means how the flowering plants reproduce sexually. In the flowering plants, the sexual mode of reproduction is having various steps, which means it is having the pre-fertilization step, and the post-fertilization step. So basically, if we are talking about the flowering plant, which means these plants are known as the angiospermic plant. So in the class 11th, you have already studied about the angiosperm. So let me brief about the angiosperm. Angiosperm are those plants which are having fruits and seeds are present inside the fruit, which means seeds are covered by the fruit. That is the basic and there is a unique feature of the angiosperm. If seeds are covered, then we can say it is angiosperm, like the guava, mango, pomegranate. They all are angiospermic plants. And these plants are having very special site of sexual reproduction that is known as flowers. So flower is the main place, main structure which ultimately involve in the sexual reproduction. So how it involves? So let me clear you. So basically flower is having various hormonal and structural changes. And these hormonal and structural changes ultimately leads to differentiation in the primordial bud, which we can say कि it is the structural changes at the time of the inflorescence. Inflorescence का मतलब होता है the arrangement of flower, right? The packaging of the flower. That is known as inflorescence. So different type of plants are having different type of inflorescence, and at this stage, these structures are having the several several structure that involve in the sexual reproduction. So typically, if we talk about the structure of a flower, typically flower has two parts, right? These two parts involved in the sexual reproduction, basically. So these two parts are the androecium and gynoecium, right? So let's move ahead. In this slide, you can see a complete structure of flower. So this is a flower, right? And in this flower you can see it is having different layers of the different part, like the sepal, petal, the androecium and gynoecium. So here there's a structure of the gynoecium and here is the structure of a androecium. So move ahead with the further explanation of the androecium and gynoecium. Now, androecium. This androecium is the male reproductive part of a flower, which means this is the male side of the flower. And if we talk about the male side, which means this flower will definitely produce the male gamete, right? So androecium. This androecium is also known as a stamen. इसको हम stamen भी कहते हैं in general. So the number of stamen, the size of a stamen varies from flower to flower, species to species. So we can say the number of stamen is not fixed for all type of flower. It varies from flower to flower, species to species. So in next slide we will see how these stamen works and what are the part of the stamen. Now the part of the stamen. Stamen, that is the male reproductive part of the flower, it is having two different parts. One is the filament and another is the anther, right? Filament and anther. So come to the part filament. Now filament. So in this diagram, in this slide, you can see this handle-like structure, this complete structure is known as filament. Okay? It is a filament. So what is filament? The filament is just a handle-like structure, and this structure is having two ends, the proximal end and the anterior end. On the anterior side, on the anterior side, it is having the anther. So this is the anther. But on the proximal side, this is the proximal side. This is the proximal side. This proximal side fix in the thalamus of the flower. So this is the thalamus of the flower, petal of the flower. So we can say this is the filament. Filament anterior part is having the different part of stamen, that is the anther, and the proximal part get fixed in the petal of a flower. Move ahead. Now anther. So anther, this is typically a bilobed structure. So if we talk about the bilobed का मतलब होता है, bilobed means two compartment. So bi means two and lobed means compartment. So this anther is having the two compartment, and each compartment, each lobe has two theca. So theca we can say in simple term is the subdivision of the lobe. So anther bilobed and each lobe two theca. So in total a typical anther has four theca, in complete four theca. So in this diagram you can see here this is the first lobe and this is the second lobe. So these are the two lobes, and each lobe, now you can see this is the first theca, second theca, and here is the first theca and second theca. So typically this is having total four theca, right? Now move ahead with further little more discussion about the anther. Now the transverse section of a anther, which means कि if we cut our anther on the transverse section, then what we will see in the transverse section of an anther, we will find the anther is a tetragonal. Tetragonal means it is having the four microsporangia. How many microsporangia? Total four microsporangia. So these four microsporangia are located at the corner of the anther. So this is the microsporangia, right? This is microsporangia, microsporangia, and microsporangia. So these are the four microsporangia sites which are present at the four corner of the anther. Now, the microsporangia further develops into a pollen sac. Pollen sac. Now, word is pollen sac. The pollen is the male gametophyte, and sac is the bag-like structure. So pollen sac means the bag, the sac that contains the pollen grain, right? So the macrosporangia that develop into the pollen sac, and it extend longitudinally all through the length of an anther. Means that this complete part, longitudinally from this to this, get filled with the pollen grain, packed with the pollen grain. Now, the structure of microsporangium. In the structure of microsporangium, we will find the typical microsporangium is nearly circular in outline, circular in structure, and each circular structure is surrounded by four wall layers: epidermis, endothecium, middle layer, and tapetum. So these are the four wall layer which present in the microsporangia. So epidermis, endothecium, middle layer, and tapetum. So in this slide you can see the outer one, this one is the epidermis, then This one is the Androecium, now middle layer, and this wall is the tapetum. So outer three wall combinedly provide the protection and help in the dehiscence of anther, means the spreading of the anther, right? But the innermost layer, that is the tapetum. This tapetum provides the nourishment for the developing pollen grain. क्या करती है? जैसे जैसे pollen grain grow करता है, this tapetum provides the nourishment to the pollen grain. Now, the cell of the tapetum. It contains a cytoplasm, and this cytoplasm is very dense, right? And generally, it contains more than one nucleus per cell of the tapetum, right? Okay. So in young anther, you can see in the young anther, means when the anther is very young, not get mature. This microsporogenum has a very special type of tissue, that is the sporogenous tissue. So this sporogenous tissue is present at the center. So this is the place where the sporogenous tissue is present, and this sporogenous tissue is compactly arranged, having the homogeneous diploid cell, and this diploid cell is known as sporogenous cell. So I think you are very much aware about the tissue is the group of cell. So if we are talking about it is the sporogenous tissue, it must contain a sporogenous cell, right? Now, so in this slide you can see the process of the formation of the pollen grain, how pollen grain form inside the anther. So as the anther develop, जैसे जैसे anther develop होता है, now each sporogenous tissue, each sporogenous cell, that is known as the PMC, pollen mother cell. क्या कहते हैं इसको? Pollen mother cell. It is also known as microspore. Microspore. So pollen mother cell or microspore, both are the same thing. It undergoes meiotic division. Very special thing, meiotic division means meiosis, which means it undergoes reduction division. So when any cell which undergo reduction division, then it convert into microspore tetrad, and these microspores are haploid in nature, because these microspores are the result of the meiosis, right? Now, the formation of the microspore from the PMC through meiosis, this is known as microsporogenesis. So what is microsporogenesis? The process of the formation of the microspore from the pollen mother cell by the process of reduction division. That is known as microsporogenesis. Fine. Now, as the anther matures and dehydrate, means get dry, then the microspore dissociate, separate from each other, and develop into a pollen grain. So this is the male gametophyte pollen grain. So in this slide you can see these are the numerous pollen grain. So roughly in each microsporangia, thousands of pollen grain are developed, right? And they're released with the dehiscence of the anther. Now the pollen grain, that is the male gametophyte. So we can say generally it is spherical in nature, ball-like structure, and 25 to 50 micrometer in diameter. 25 to 50 micrometer in diameter. And very special thing, this pollen grain is having two layer wall, exine and intine. So you can see this is the outer layer which is having the hook-like structure. It is the exine, and inner layer it is the intine. So the basic difference between the exine and intine, exine is a hard coating which is made up of sporopollenin, and it protect the anther from the adverse condition. This exine is present all over the pollen grain except its aperture, that is known as germ pore. Now, intine, it is the inner wall, thin, continuous, made up of cellulose and pectin. You have to keep in mind. So this is about the male part of the flower, the development and formation of the male gametophyte. Thank you.